In April 1955, in the United States, parents did something they had never done before. They lined up to give their children a shot, not because they had to, because they wanted to, desperately. Polio had stalked the country for decades. Every summer, the same ritual of fear, pools closed, movie theaters shut, children kept indoors. By 1952, nearly 58,000 Americans caught it in a single year. Thousands died. Thousands more never walked again. Some ended up in iron lungs, steel cylinders that breathed for them because their own bodies no longer could. Then Jonas Salk announced a vaccine. The trials worked. On April 12, 1955, it was declared safe and effective. Church bells rang. Scientists cried in press conference rooms. Parents exhaled for the first time in years. 13 days later, a child who had just received the shot was admitted to hospital with polio, then another, then more. This is the story of the vaccine that gave children the disease it was supposed to prevent. Poliomyelitis, infantile paralysis, was invisible. It spread through contaminated water and surfaces, and most people who caught it had mild symptoms or none at all. But in a small percentage of cases, the virus crossed into the nervous system and destroyed the motor neurons controlling muscles. Within hours, a healthy child could lose the use of their legs or their arms or stop breathing entirely. There were no patterns parents could avoid. It struck clean neighborhoods and dirty ones, rich families and poor ones. Franklin Roosevelt, President of the United States, had been paralyzed by polio at the age of 39. By 1952, the worst epidemic in American history hit. Nearly 58,000 cases were reported. More than 3,000 Americans died. Around 21,000 were left with some form of paralysis. Hospitals overflowed. Swimming pools and movie theaters closed. Parents kept children inside all summer, terrified of the invisible enemy. The March of Dimes, a nationwide fundraising organization, collected millions of dollars to fight the disease. Jonas Salk, a virologist at the University of Pittsburgh, was working on a vaccine. By 1954, the largest medical trial in history began. Nearly 2 million American children, called polio pioneers, received either the vaccine or a placebo. On April 12, 1955, the results were announced. The Salk vaccine was declared safe, potent, and effective. Church bells rang across the country. It was one of the greatest victories in the history of medicine. But what came next turned that victory into a nightmare. Before we get to the disaster, let's understand what the Salk vaccine actually was. Polio is caused by a virus, a very small, very tough pathogen that attacks the nervous system. The Salk vaccine was called an inactivated vaccine. The idea is simple. Take live polio virus, kill it completely using formaldehyde, and inject it into the body. The immune system sees the dead virus, learns to recognize it, and builds antibodies. Later, if a real polio virus shows up, the body is ready. The critical word is completely. The formaldehyde had to kill every single virus particle. A single surviving live particle could replicate inside a child's body and cause the very disease the vaccine was meant to prevent. Salk knew this. The manufacturing process was detailed and demanding. The virus had to pass through filters of specific pore sizes, be inactivated for the right duration, and tested at multiple checkpoints. Lab animals received the preparation first. The process worked. Multiple manufacturers had already shown it could be done safely. There were 6 licensed manufacturers in the United States. Each was given the same set of protocols. Each underwent federal inspection before release. One of those manufacturers was Cutter Laboratories, a mid-sized company based in Berkeley, California. They had less experience producing complex biological preparations than some of their competitors, but they were licensed. They had passed the inspections, and in April 1955, they released their batches. The nationwide vaccination campaign began immediately after the April 12th announcement. Within days, millions of doses were flowing to clinics across America. Parents brought their children in. Nurses gave shots. Everyone believed the nightmare was finally over. Then, on April 25th, 13 days after the campaign began, a call came into the California Department of Public Health. A child vaccinated with a Cutter dose had developed polio. Then another call. Then more. By the end of April, the pattern was undeniable. Children who had received Cutter brand vaccine were getting sick, and the paralysis was striking the arm that received the injection first. That was the signature of vaccine-induced infection. The virus didn't spread randomly through the body. It started at the injection site. The federal government suspended the entire vaccination program on May 7, 1955, just 25 days after its triumphant launch. Cutter recalled its batches. Investigators quickly identified several failures, all converging at Cutter. The inactivation process had not completely killed the virus. Among the 3 types of polio virus used in the vaccine, Cutter's preparation included a particularly virulent strain, type 1, that was harder to inactivate and more dangerous if it survived. The filters used in Cutter's process allowed clumps of virus to pass through, shielding particles from full exposure to formaldehyde. And the safety tests used to check for live virus were not sensitive enough to catch the small amounts that survived. Critically, Cutter had formally followed the written protocols, but they lacked the production expertise and process control that more experienced manufacturers had developed through practice. The same instructions, in less experienced hands, produced a deadly product. Around 200,000 children received Cutter batches containing live polio virus. They did not know. Their parents did not know. And the system that was supposed to prevent this had missed it entirely. The numbers that emerged from the investigation were precise and devastating. According to the analysis by epidemiologists Neil Nathanson and Alexander Langmuir, published in 1963, approximately 40,000 cases of polio were caused by Cutter vaccine. Around 200 children were left with some degree of permanent paralysis. Approximately 10 children died. But the spread did not stop there. Because the children who were vaccinated could shed the live virus, they passed it to siblings and classmates. The secondary cases had an even higher rate of paralysis, because those people had no immune protection at all. The investigation by Nathanson and Langmuir was methodologically rigorous. They compared case rates across all manufacturers and found a clear, unmistakable signal. Paralysis was linked specifically to Cutter's high-risk batches. This wasn't a systemic failure of Salk's vaccine. It was a failure at one company. For the families involved, that distinction meant very little. Anne Gottstanker was 5 years old when she received a Cutter dose. She developed paralysis. Her family sued. The trial was a landmark. The judge found that Cutter had not been negligent. They had followed the written protocols, but still ruled against them under an implied warranty of safety. The product was supposed to be safe. It was not. The company was liable. That verdict would reshape the legal and regulatory landscape for vaccines for decades. The Cutter incident did not destroy confidence in the Salk vaccine permanently, though it came close. After the recalled batches were identified and removed, the vaccination campaign resumed within months. By 1957, polio cases in the United States had dropped by 90%. By 1979, wild polio virus was eradicated from the country entirely. But the disaster triggered a transformation in how the United States, and eventually the world, regulated vaccines and pharmaceuticals. Before 1955, federal oversight of vaccines was limited. The Division of Biologic Standards, which had licensed the manufacturers, had relied heavily on self-reported data and standardized written protocols. The assumption was that if a manufacturer followed the instructions, the product would be safe. The Cutter incident proved that assumption was wrong. In the years that followed, federal regulators demanded lot-by-lot testing. Every single batch of vaccine produced had to be independently tested for live virus before it could be released. Not just by the manufacturer, but by federal labs. The sensitivity of those tests was dramatically upgraded. Process controls became more specific, not just filter the virus, but which filter, at which pressure, inspected how often. The regulatory authority of the Division of Biologic Standards was expanded. It eventually became part of the FDA's Center for Biologics Evaluation and Research, the body that today reviews every vaccine, every blood product, every cell line before it reaches a patient. Paul Offit, a vaccine expert and historian of the Cutter incident, wrote that as a direct result of the disaster, the safety standards for vaccines became arguably unparalleled among medical products. The very system that makes modern vaccines extraordinarily safe was built in large part on the lessons learned from what went wrong. The Cutter incident is not a story about vaccines being dangerous. Every vaccine used today went through a regulatory gauntlet that was built, in part, because of what happened in 1955. That system is imperfect, but it is incomparably more rigorous than what existed before.